All right. Now we're going to talk about the electron transport chain and where we produce the majority of the energy through aerobic respiration. There are two basic processes that will occur. We're going to release the electrons and the hydrogen ions from the NADH and the FADH. So we oxidize those molecules. And then we're going to produce a lot of ATP, and in the process, we will produce water as a waste product. So we've produced a ton of carriers, coenzyme carriers. In glycolysis, we produce 2 NADH. In transition, we produced 2 NADH. And then in citric acid cycle, we produce 6 NADH and 2 FADH. So a total of 10 NADH and 2 FADH were produced. And so in the electron transport chain, these electrons are going to be removed from the NADH and the FADH, and they're going to be shuffled from one carrier to another carrier. In this process, they release energy. This energy that they release actually allows us to push the electrons from one side of the membrane to the other side of the membrane. And you're not seeing it here. Let me get here. It's a good picture. So here we have NADH being oxidized to NAD. Electrons are going to be passed from one carrier to another carrier to another carrier to another carrier. And the hydrogen ions are pushed across the membrane from a lower energy to a higher energy environment. So we produce this high energy ion gradient. And just like anything, ions want to move from where they are to where they're not so that they're in equilibrium. In our cells, this process occurs in the cristae of our mitochondria, but in bacteria it occurs right in the cell membrane from one side of the membrane to the other side of the membrane. And so we have this ion gradient. These ions want to move from the higher to lower gradient. And so they're going to flow through the one molecule, the one protein that allows them to. They flow from higher to lower concentration down through an ATP synthase molecule. And as they do that, they activate the protein. The protein spins and produces ATP from ADP. The final electron acceptor is oxygen. Oxygen binds to the electrons, and so when the hydrogen fly through this ATP synthase molecule, they are attracted to the negatively charged oxygen and attach, producing water as a byproduct. And you see this here. Oxygen binds, and then the hydrogen moves down, and it's also going to bind, to the water, producing H2O as one of the end products of this process. And so we end up having, another thing I want to show you. Here we have the first complex. Here we have another protein. So we're looking at the proteins that can pump hydrogen across. And there's the third protein. NADH drops its hydrogens off on the first protein. FADH drops its hydrogens off on the second protein. And because the hydrogens have a smaller area to move across, they don't produce as much ATP, which is why for every FADH that drops their hydrogens off and electrons, they only produce 2 ATP giving us 4 ATP. The rest, NADHs, drop off. They produce about 3 ATP per. So that's where we're getting this 6 times 3, 2 times 3, 2 times 3 is where we're getting these numbers. So the end process, we make about 36 to 38 ATP for the complete oxidation of one glucose molecule. This is a great song you can listen to. I'm not going to listen to it with you right now, but it's a really fun song. If you like the Black Eyed Peas, you'll love it. We can use other molecules to break down and produce energy. Glucose just happens to be the favorite fuel, and we typically use glucose for everything. That doesn't mean that we have to use glucose. So if we don't have glucose available, our body will use proteins or fats just as easily. And so here we have amino acids. If we remove the nitrogenous region, the nitrogen containing portion of the amino acid, then we can, and that's termed deamination, we can move these amino acids into different regions of the aerobic process. If we're using fats, glycerol can enter directly into the production of glyceraldehyde 3-phosphate, since glycerol is part of that molecule. And the fatty acids can go right into the production of acetyl-CoA. We have to, in this case, break down using beta oxidation to cleave one acetyl group from the fatty acid at a time. That will then enter and attach to coenzyme A and move through the citric acid cycle. I've already mentioned these, so I'm not going to talk about this again. We do have anaerobic processes that also produce energy. So you can use anaerobic respiration, where we use other final electrons, not oxygen. And this does produce ATP, but a very small amount of ATP. And then we have fermentation. I don't like that. Where we use anaerobic means to produce energy, but we only follow that glycolytic pathway. And so here you can see what happens in fermentation. We have this pyruvate at the end of glycolysis, but because we need to produce more NAD so that we can oxidize glucose again, NADH is oxidized to NAD, and pyruvate is reduced to lactate. So this is what some cells do. All animal cells do this, and some bacterial cells also use this process. And we produce lactic acid as a waste product. And then other bacteria use, or in a fermentative process, produce alcohol and carbon dioxide. So here again, we have pyruvate, we release carbon dioxide, and we produce ethanol when we oxidize the NADH back to NAD. And so here are some products associated with fermentation: ethanol, alcohol. Propionic acid. Propionic acid is used in the production of different foods. Mixed acids. This is one way we can actually identified different species of bacteria. And so that's the end of fermentation. I'm sorry, I didn't have a lot for fermentation. This is a song that talks about fermentation. I'll let you listen to that song on your own. And we will go through this in lab on Thursday just to make sure you understand what's going on, okay? The last process we're going to talk about then is photosynthesis. Photosynthesis is the production of a carbohydrate or the buildup of material from the use of or from sunlight energy. So sunlight energy is the ultimate form of energy, and photosynthetic organisms trap sunlight energy and convert it to the energy of a carbohydrate or an organic compound. And they can use that to either produce ATP or to build up cell walls, proteins, other things. In eukaryotes, this process occurs in the chloroplast, and we have molecules called chlorophylls that take in sunlight energy. In prokaryotic organisms, this process occurs in the cell membranes, and they also have pigment absorbing molecules, but they're not always the same. Depending on the species, they're going to be slightly different in molecules. Photosynthesis is divided into two sets of reactions. We have the light reactions, and then we have the dark reactions. Light reactions are energy fixing reactions where we take in sunlight energy and then we produce everything we need for the dark or carbon fixing reactions. And then the carbon fixing reactions are where we actually produce the organic molecules. So light dependent or light fixing reactions. We have two basic photosystems associated with this. We have photosystem 1 and photosystem 2. They are identified by, let's see, how were they identified? Photosystem 2 is the first one we use, by how they were actually found. So we found photosystem 1 first, and then we found photosystem 2. But photosystem 2 is the first photosystem in the non-cyclic pathway, whereas photosystem 1 is found in both cyclic photophosphorylation as well as in the non-cyclic pathway. It's the second photosystem. And so we identify them by how we identified them, but not how they work. That's what I want to say. Cyclic photophosphorylation uses only photosystem 1, and it produces a lot of ATP, but does not produce any reducing molecules. And so in this case, the reducing molecules are going to be NADPH. Anytime we're building something, we use NADP for the anabolic pathways, whereas we use NAD and FAD for catabolic pathways, breaking stuff down. In the non-cyclic photophosphorylation, we produce ATP, but we also produce NADPH. So here is non-cyclic photophosphorylation, and we can also see and we can see the cyclic photophosphorylation. So here we have photosystem 2. Takes in the rays of sunlight, and it activates the electrons. So it energizes the photosystem. In aerobic photosynthesis, water is split. Oxygen is released. the electrons and the hydrogen ions then move into photosystem. The electrons move into the photosystem 2, and then the hydrogen ions are going to be pumped across a hydrogen ion gradient, or pumped across a membrane producing a hydrogen ion gradient. This will occur in the plasma membrane of the cells again. So as the electron moves into photosystem 1, the energy activates that electron and causes the electron to get more and more excited. And it moves up in excitement levels until it gets so excited it jumps and moves down electron excitement levels from one carrier to the next. This is what drives the pumping of the protons across the membrane. And the low energy electron moves into the reaction center of photosystem 1. We get another beam of light that activates that electron again. The electron gets very excited again. And in non-cyclic photophosphorylation, it will jump and attach to NADP. We have these hydrogen ions that are pumped across a membrane. They're in a high hydrogen ion gradient, and they want to move back across to the low hydrogen ion concentration. So they're going to move across an ATP synthase molecule. And when they do this, they activate the ATP synthase, just like in aerobic respiration, and we produce ATP. But at the same time, the hydrogen moves across, and it's going to attach to the NADP as well. As the electrons attach, hydrogen ions are going to be attracted to the negative NADP, and we produce NADPH. If this is cyclic photophosphorylation, we're only using photosystem 1. In that case, the electron that enters the reaction center of photosystem 1 gets activated, moves up in excitement levels, and then immediately jumps down, activating the electron transport chain to produce more ATP, to drive hydrogen ions across the membrane, and then have the hydrogen ions go back across through the ATP synthase. So we constantly cycle those hydrogen ions, and that's and we're cycling the electrons, and that is cyclic photophosphorylation. So in this process, we don't produce any reducing power of NADPH. We just produce energy, and this is important because in certain photosystems, we need more energy to produce carbohydrates. So we have to be able to produce energy even if we don't need the reducing power. So here's a video that I'll play for you.
In bacterial photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes down through an electron transport system and finally back to the reaction center. The energy released during this electron transport is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation. No reducing power needed for biosynthesis is generated in this process. Plants and cyanobacteria utilize two photosystems which work sequentially to produce both energy and reducing power. First, a photon of light ejects a high-energy electron from photosystem 2. The electron lost from photosystem 2 does not return to photosystem 2, but is replaced by an electron generated from the enzymatic splitting of water and the release of oxygen. The electron then travels from the excited reaction center of photosystem 2 down an electron transport chain and finally to the reaction center of photosystem 1. This electron transport system generates a proton motive force that is used to produce ATP. Since the excited electron does not return to photosystem 2, this mechanism for making ATP is called non-cyclic photophosphorylation. When photosystem 1 absorbs a photon of light, it ejects a high-energy electron, which is used to drive the formation of reducing power in the form of NADPH. The ejected electron is replaced by an electron from photosystem 2.
All right, I'm going to stop it here. So in anaerobic bacteria, we can't split water to produce the oxygen. So we use other molecules like hydrogen gas or hydrogen sulfide to produce the electrons and the hydrogen ions. And we don't produce near the amount of energy as we would in aerobic mechanisms. Because of this, we're going to have to use extra energy. We'll have to produce extra energy just to run the photosystems. Purple bacteria are anaerobic, and they have to use extra energy to reduce NAD or to reduce NADP. In green bacteria, they're also photosynthetic. They generate or they have to produce a lot more ATP through the cyclic photophosphorylation so that they can then reduce carbon dioxide to a carbohydrate. So the next process is carbon fixation. So in this process, we use carbon dioxide, which we receive from the air typically, and we convert that carbon dioxide into a carbohydrate. Typically, we think of glucose as the carbohydrate that we're looking at. A lot of times we look at glyceraldehyde 3-phosphate. That's what's actually produced with every turn of the Calvin cycle. The first process is going to be fixing the carbon dioxide, which is a 1-carbon molecule, and we fix it to a 5-carbon molecule that immediately becomes unstable and breaks down into 2 3-carbon molecules. So here we have, so let's just look at it this way. 1 carbon dioxide comes in, attaches to a 5-carbon RUBP molecule. This 5-carbon RUBP molecule turns into a 6-carbon molecule, which is unstable, and immediately breaks down into 2 3-carbon phosphoglycerates. Phosphoglycerate is then converted through reduction to glyceraldehyde 3-phosphates. So for every 3 that come in, we produce 3 glyceraldehyde 3-phosphates. Do you see that? Now we need to have the cycle run 6 times so that we can produce our glucose molecules. Because for every 6 glyceraldehyde 3-phosphates we produce, we only get to gain 1 glyceraldehyde 3-phosphate. The rest have to be used to make up more RUBP, which is needed since this is a cyclic activity. And we're going to use ATP. We're going to oxidize the NADPH and reduce the carbon dioxide. Oh, hello. Okay. So now let's watch this video.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
So that means it has to turn 6 times because glucose is a 6-carbon molecule. Okay. So we have a microorganisms, I shouldn't say we, but we do too. We have very good metabolic diversity. Some microorganisms are autotrophs while others are heterotrophs. Autotrophic organisms are organisms that can produce their own food. If you're a photoautotroph, you use light to produce the energy, and you produce food from carbohydrates. From carbon dioxide, inorganic carbon dioxide. If you're a chemoautotroph, you use inorganic compounds as energy, and you produce food from inorganic carbon dioxide. Heterotrophic organisms gain energy from some organic molecule. If you're a photoheterotroph, you use light as your energy, and you use an organic molecule as your carbon source. Chemoheterotrophs, which is what we are, use organic compounds for both energy and carbon dioxide, and carbon, not carbon dioxide, and our carbon source. We have saprobes, which are organisms that use dead organic matter for their source of carbon and energy, and parasites, which can only use living tissue as organic matter for their nutrients and energy. And that's it for this chapter. So I'm going to shut it down and upload this video. Have a great day.